Okay, so that was DNA structure and basically how we figured it out. And so now we come to DNA replication. How do you make more copies of DNA? So, again, resemblance of offspring to parents, like in the pea plants or any other organism, the offspring look like parents or tend to. And so that relies on accurate replication of DNA prior to meiosis and its transmission to the next generation. So replication prior to mitosis also ensures, the faithful transmission of genetic information from a parent cell to the two daughter cells. So either way, whether it's for meiosis or mitosis, basically, it's important to accurately copy DNA so you can carry on accurate copies of the DNA to either the new somatic cells that will then end up dividing again and again to become more somatic cells. You want to make sure that the DNA is correctly copied and correctly passed on. Or in meiosis, you also want to make sure that they are going to get correctly copied and correctly passed on, so that this next generation, can have the right copies. So again, Watson and Crick noted that the specific base pairing suggested a possible copying mechanism for genetic material. So now, we know more about that, and the copying of DNA is called DNA replication. So since the two strands of DNA are complementary, we always know A binds with T, G always binds with C. So each strand kind of acts like a template for building a new strand. So when you need to build a new strand, you unzip this one, and that helps you build the next half of each one, and you end up with two identical molecules. So when you do this, the yield is exactly two replicas of the parental molecule. So here is a figure showing that better than just my fingers. So here is the parental molecule. And so DNA is anti-parallel, so actually it would be like that. So if one strand is going this way, the other strand is going that way. And so to also talk more about the anti-parallelness of strands, so one end of a strand is called the 5' end, and then the other end of that strand is called the 3' end, and that is based on what is sticking out in the sugar phosphate backbone. So let's go back here. Okay, so these are the nucleotides, right? And so sticking out of the sugar, there are so shown in pink are the carbons in this sugar. So again, in organic chemistry, we kind of do shorthand, and so this is a carbon. It's just implied because there's just a point. You don't even need to put a C there. We know that's a carbon. And so these little pink ones, these pink numbers are showing the numbering in the carbons, and you kind of go clockwise. And so if this is carbon 1, the 1' carbon, the 2' carbon, the 3' carbon, 4' carbon, and then actually there's another carbon that sticks out right here. So there's 5 carbons in this molecule, the sugar. And so stuck to this 5' carbon is the sugar phosphate backbone. And so based on that sticking out, we call this the 5' end. And then at the other end of the strand, there is going to be another nucleotide connected here to the 3' carbon. So anytime you stack on another nucleotide, you do that here. You connect it to the 3' carbon on the ribose or deoxyribose. And so this end is the 3' end. So anytime you're adding more nucleotides, you put a carbon or you connect, the phosphate group to there. So yeah, that's why we call it one end is the 5' end, and then one end is the 3' end. So now if we come back, hopefully the anti-parallel thing makes more sense. So if one is going this way, the other one has to go anti-parallel like that. And so when you unzip them, you basically just look at what letters on this side, and then you put the nucleotide that goes with this one. So it's kind of like a fun puzzle, I tell students. So if there's an A, you would put a T here. If there's a C, you would put a G here, because remember those rules. A always binds with T. G always binds with C. So that's how you would build the next half of this strand. And then the same with this other strand. So if this is a T, you put an A here. If this is a G, you put a C here. And then by the end, lo and behold, you have two molecules that are actually identical to each other. And each one has half of the original parental molecule, the darker blue. And then the newer one that you built is the lighter blue. And so that is what's called the semi-conservative model of replication. They were trying to figure out how DNA gets made. Do they just take a DNA molecule and build a whole new DNA molecule? Do they split it in half to help build the new ones? Are there just pieces of the parental molecule mixed in with the new stuff? And so this is what's called the semi-conservative model that we know is true now. So this semi-conservative model of replication predicts that when a double helix replicates, each daughter molecule will have one old strand, so the parental one, and then one new strand. So that's why it's kind of conserved when the old strand is conserved from the parent molecule. And then there's also the new strand. So that's the semi-conservative model. There were other models too. So there was the conservative model where it's just like the parent strands just rejoin. And then there's a whole new molecule of two new strands that are together. That was the conservative model. Like the parent ones just stay conserved. And then there's also the dispersive model that each strand is just like a mix of old and new stuff. And so that's what this is. So this was the conservative one, like the parent cell unzips, you build each half, and then maybe those new things zip together. And then the old thing stays conserved together. The semi-conservative model is it unzips, and then they kind of stay like that, like the new half stays with one parental half, and so it's semi-conserved. And then there's the dispersive model where they thought, well, maybe just parts of the old one get mixed in with new stuff. And then that's not the case. So now we know it's the semi-conservative model because of more experiments. So these experiments by Matthew Mieselson and Franklin Stahl supported that semi-conservative model. And all these experiments, sometimes it's kind of like a drag to learn about, but if you reframe it, it's really interesting how people got creative to get to know the things that we now know and kind of take for granted. But back then, you didn't know these things, and so you had to kind of make really creative experiments to show things. So yeah, it's also interesting. Try to tap into that maybe. So here they had bacteria that were cultured in medium, or like just the stuff that you grow cells in is called media. And so here's one medium that has a heavy isotope of nitrogen. And then here are bacteria that had media with a lighter isotope of nitrogen. So remember isotopes from back in chapter 3. And so they took bacteria cultured in media with the heavy isotopes and transferred, and the bacteria transferred to medium with the lighter isotope. And then again, they put them in a test tube and centrifuged and span it really fast. And so this separated things based on how dense it is. So things that are more dense tend to fall towards the bottom. And then things that are less dense tend to like separate out further up. And so when DNA was centrifuged after the first round of replication, they found everything in one spot. But then when they centrifuged it again after enough time that they knew, okay, a second round of replication happened, because there's this much more cells, then DNA replication must have happened. And so if we centrifuge it again, it turns out that they separate. And so based on the bases that they used that were available at the time of replication, so either the heavy or the light, that is going to show different like separations, different patterns of separation in the test tubes based on, like when did they do the replication and what did they use? And then how does it look afterwards? So they compared, those samples before and after replication. And so they looked and they saw this pattern. Okay, if we spin it like this, this is where the layer with our DNA is. So that means there's some old and some new. And then if they look at it again after it was centrifuged, after the second round of replication, this is the pattern they saw. And then this helped show, okay, this is the semi-conservative model. Because this is how heavy it was, because it's a mix of the heavier and lighter. So it ends up in a medium. And then the lighter stuff ends up here. It's not like a bunch of pieces that are mixed. It's not like, oh, there's only one heavy part and then there's a bunch of like... So this is that combination of the heavy and light that ends up in the middle in both rounds that showed it's semi-conservative. I should have placed this earlier. Maybe not. So this is for you, another worksheet to help demonstrate and act out and physically draw DNA replication to kind of help you get the picture. So this is just kind of, a little drawing cartoon of a molecule of DNA and then of nucleotides and the, anti-parallel strands. And so, you could color them in, color the phosphate groups, yellow to follow the figure. I think the deoxyribose was blue, and then whatever base, whatever. But, notice that A and T always bond together in whichever orientation, and then there's 2 double bonds between A and T. And then G and C always bond together, and there's always 3 hydrogen bonds. And so that is, DNA structure. And then to kind of help you show and act out replication, so here is the parental molecule, and you unzip it, and then based on what's here, you can write the corresponding letter. So if we did this together, this is a T, so this would be an A. Again, A and T always bind together. So here's a reminder. G and C always bind together. So if this is an A, this one's a T. If this one's a C, this one's a G. And then... And, I have it hammered in, but, it might take you a little bit. Just remember, and by physically doing this, it does help you remember, okay, C, G, A equals to T, A equals to T. And so if you did the same for the bottom strand, by the end, you should see they should be identical to each other. And so that is how you make 2 molecules that end up being identical to each other. And then remember, it's the semi-conservative model. So if you wanted to do, add some more detail by coloring in, one strand, the parental strand with dark blue, and then maybe it helps to know, okay, this parental strand, dark blue. And then do the other strand light blue. So what you're building new is going to be done in light blue. And then that also helps demonstrate the semi-conservative model of replication. And same for this one. And then so at the end, it makes two identical DNA molecules. Even though we're showing the color of like dark blue and light blue to show the parental strand and the new strand, like everything else is going to be identical, you know? And then I'm just going to shorten this. So you can either draw it or print it. I think it really, really does help to get that physical practice in. And so I like giving these worksheets because it's a good overview and you get the big picture first, and then you can add in all these other details. So, you know, DNA replication, a closer look in more detail. So the copying of DNA is pretty remarkable in its speed and accuracy. And so not shown in that picture, but there's actually like more than a dozen enzymes and other proteins that are part of DNA replication. And so, yeah, there's all these details that you can add after to that worksheet to help show different things. And so replication in bacteria is best understood, but evidence suggests that the replication process in eukaryotes and other prokaryotes is, you know, fundamentally similar. So DNA replication starts at these sites called origins of replication. So where it like unzips, we call the origin of replication. So when they're both opened up, this makes like a replication bubble. So as mentioned in part 1, there are different parts where DNA is replicated because again, DNA is really, really, really long. And so it actually helps to start replication at multiple parts, and then you could kind of just fuse the new pieces together instead of totally unzipping. And so in that way also, this worksheet is kind of helping just demonstrate the semi-conservative model, but this isn't exactly how it's done. You're not just going once and then going all the way down. So that is, you know, also a disclaimer. The worksheet can't be super detailed because it's supposed to introduce the concept and then you can add all this. So in one long section, there's actually multiple places where replication is happening. And so in one area, you unzip at the origin of replication, and then there is one on the opposite side, and then here is like a bubble that gets made. And so, especially because eukaryotic chromosomes are so big and so long, a eukaryotic chromosome may have hundreds or even thousands of origins of replication. And then replication proceeds in both directions. So it starts building this way and then this way until the entire molecule is copied. So here's a picture showing that. On the left is replication in an E. coli cell, and so their chromosomes are circular, and so they just kind of start at one point and then unzip a little bit and then build in the whole direction until you have two circles. Yeah, so they have an origin of replication here, and they really just need to do one bubble, because then eventually it just grows and then there's two. On panel B, on the right-hand side, are eukaryotic chromosomes. And so they're super long. You know, this isn't even showing the whole thing. This is just a section of it. But so here is a chromosome. Here is one origin of replication. and then there's a couple others, and so that's where it unzips. And then where the parental strand in dark blue unzips and forms the bubble, you start building the new strand that's shown in like the light blue. And so as you build more and more, you add more nucleotides, the bubble gets bigger, and then eventually the bubbles like intersect, and then you just fuse the new strands together. And so that's how you end up with 2 total DNA molecules with the new daughter strand, and then also the parental strand. They're like all connected. It's not just like a bunch of chopped up pieces. Again, this is a semi-conserved model. And yeah, so we know this because we've seen it. This is what it looks like with 1 chromosome and multiple origins of replication. And so at the end of each bubble is a replication fork. So if we go back, these are replication forks where it like forks off from each other. You know, there's like a fork in the road. But at the end of each replication bubble is a replication fork, a Y-shaped region where the parental strands are being unwound or unzipped. Excuse me. And so there are enzymes that are in charge of doing this, and they are called helicases. They are enzymes that untwist the double helix at the replication forks. It's like unzipping and then like untwisting. And there's a bunch more, guys. So, yeah. So helicases, you know, unzip it. Single strand binding proteins bind to and stabilize those now single-stranded DNA molecules. So once they get unzipped, it's like a single strand, right? And then so these single-strand binding proteins bind to them and help stabilize them, kind of prevent it from like zipping back together. It holds it open. There is another enzyme called topoisomerase. And so this enzyme helps relieve the strain of twisting. Because again, like the DNA double helix is twisted because it kind of has to be like the physical conformation of those molecules and atoms and how they have to stick out, like it twists for a reason. And so topoisomerase helps relieve the strain of this twisting and breaking. This twisting by breaking, swiveling, and rejoining DNA strands. So it kind of holds it steady. I'm trying to think of an analogy, but I can't, so whatever. So here is a small summary of these few things. So the first one, again, helicase is kind of like the zipper, like unzipping something. So helicase unzips it. The single strand binding proteins are proteins, and they just bind to the single strand of DNA to help, help stabilize it, keep it from like rezipping. Topoisomerase, again, helps like relieve this strain of it wanting to twist back into the helix by breaking and twisting things and helping like relieve that. And then we will talk about this guy in a second. So what you built when you were doing the worksheet, hopefully, as you were looking at what letters there, and you're like, okay, that's an A, I have to put a T here. This is a C, I have to put a G here. So you were basically acting out the job of this enzyme called DNA polymerase. And that is the enzyme in charge of polymerizing DNA. So it's building DNA by taking all the nucleotides and putting it, together. But those guys need a primer to help know to start, basically. So DNA polymerases require a primer to which they can add nucleotides. So there has to be like some starting material there. And then it knows to start like adding on stuff. So that is what is shown here. This primer is actually RNA. It's temporary. Again, it just has to be there so that DNA polymerase can start adding stuff to that end. So DNA polymerases need that primer there, and so there's another enzyme whose job it is to put the primer there. So this is synthesized by the enzyme called primase. It puts down that short RNA primer. The primer is about 5 to 10 nucleotides long. It is really short, but it's necessary for DNA polymerase to start being able to add things. So that new DNA strand will start from the 3' end of that RNA primer right here, and then you add stuff on. So remember, 5 to 3 is based on that carbon and what's sticking out. So the phosphate group is sticking out from the 5' end, and then you would add the new phosphate to the 3' end. Those are 2 more players. The primase has to put down a primer. So enzymes called DNA polymerases are what catalyze the synthesis of new DNA at that replication fork. They read what's there and then they put the right nucleotide. They're like making new bonds as they do all this. So most DNA polymerases require a primer and a DNA template, which makes sense. Like, you need to have something to read to be able to start building it. And so this guy works pretty fast, actually, DNA polymerase. The rate of elongation, like building this, is about 500 nucleotides per second in bacteria. So it's super fast in bacteria. And then about 50 per second in human cells, which is still pretty, So each nucleotide that DNA polymerase adds on is added to a growing DNA strand, and each nucleotide at first is a nucleoside triphosphate. So let me show a picture here. So this is a nucleotide that's kind of like loose in the cell, right? So it's a modified nucleotide. It has 3 phosphate groups. Doesn't this look familiar? Like ATP? Yeah, ATP is a modified nucleotide or nucleoside. Okay, so each nucleotide that is added is a nucleoside triphosphate. And so DATP supplies adenine to ATP. So this is kind of what helps show that there is a deoxyribose attached to an ATP. So DATP supplies adenine to DNA, and it is similar to the ATP of energy metabolism. The difference is just their sugars. So DATP has deoxyribose, and ATP has ribose. So yeah, ATP is literally just like a modified nucleotide. It just has a different sugar there. It's a ribose instead of deoxyribose. And so that's what D of DATP stands for. So yeah, that's what I was saying. Of like, doesn't this look familiar? Like, it kind of is. Yeah. So as each monomer, each nucleotide joins the DNA strand, that is done by a dehydration reaction. So again, if you wanted to look back at chapter 5 about dehydration reactions, how those work, that might help. So as each monomer joins the DNA strand, there's a dehydration reaction happening that's adding it on. And so it loses 2 phosphate groups as a molecule of pyrophosphate. So here is a nucleoside triphosphate, right? And so basically those 2 phosphate groups get like chopped off and then it gets added on.